Thank you. Sorry. Hi, my name is Chia Liew, and I'm a bioinformatician at the Bioinformatics core at UNL. And today I'm going to talk about Porcine Reproductive and Respiratory syndrome virus and how its infection upregulates negative immune regulators and T cell exhaustion markers. So this work was done and published together with Dr. Liew at the Department of Animal Science at UNL, and it was published last year at Journal of Virology. So you can access the paper with the QR code on the right-hand side. Yeah, feel free to take a look. There is a lot more about virology and immunology that is kind of beyond me because I'm a bioinformatician. So if you're interested in the details of like slides, staining, kind of like the putative genes that we identify through the results of the study, and there's a lot more there if you're interested in that kind of details. So PRSV PRRS is an envelope virus. Hold on, can I see like my notes somewhere on the thing? Do I just change the view? Sorry.
Do you want
the speaker view with like the two monitors?
It will also be seen here.
Oh,
Yeah.
Really?
Yeah, sadly.
Oh, okay.
Is that fine?
It's okay. I'll try my best. Thank you. Okay, so let's try again. So PRRS is an envelope positive sense and single-stranded RNA virus. It is very similar to SARS-CoV-2 in the sense that SARS-CoV-2 is also envelope positive sense and single-stranded RNA virus. It's just that it's a lot bigger than PRRS. PRRS has only like the genome size half of the SARS-CoV-2, and they belong. Well, PRRS belongs to the nidovirus order, the family Upregulates, and the genus Porcine. And that, the picture on the right is a picture of PRRS, if you're interested. PRRS is highly host and tissue restricted. It only infects pig, and one of the primary target cells of the virus is the pulmonary alveolar macrophage of the pig. Once the transmission to the upper respiratory system is established, it would first infect lymphoid tissues, but after that, it will infect the pulmonary and intravascular alveolar macrophages of the pig, and then it would cause interstitial pneumonia, and it would also make the pig or the host more susceptible to pathogens in the future, so then would increase the probability of pig getting respiratory illnesses in the future. So the rationale for our study was previous studies often study the PEM cells of pig for PRRS in vitro, and also just using bulk sequencing cells or bulk RNA sequencing but we think that because since PEM cells are only infected in very small percentage of like less than 2%, it doesn't make sense to look at bulk RNA-seq, because then we're just looking at everything, like every cells that is infected or not infected. So we adapt a different approach of doing it in vivo. So we infect the piglets with PRRS, and then we are trying to evaluate the PEM cells that are infected and non-infected among all the PEM cells. And we are also interested in figuring out how PRRS has been evading the host immune system. So our experimental design is kind of summarized in this picture. So we first take 4-week-old piglet, and then 7 days post-infection, we would extract the lung tissues of the piglet, and then we selected for cells that express CD163 plus, which is the main cellular receptor that the virus used to get into the host cells. And then we sort the cells into the bystanders and PERS-infected cells using flow cytometry. So this was done during the acute infection stage of the pig, and also we use a strain of PERS that has GFP plus infected embedded, and so that we can use this technique to sort them into PERS infected and bystander cells. I kind of added little green dots there to kind of show that the GFP plus is the one that we is that express the PERS, the GFP embedded PERS. So here is the results of our study. So we found that, so this is just, okay, I'm also going to show that. This is just like a simple alignment against the genome of PERS. And we can see that the GFP plus cells has a lot more viral rates compared to the GFP minus and the MOC cells, which are just control pigs that were non-infected. And it's also good to see that for the GFP minus, there is nothing where the eGFP is. If you look at like the GTF or like the G model there, so which means that our experiment was successful and it worked the way we intended it for. Most rates align to the 3' end of the viral genomes, where a lot of subgenomic mRNAs for structural genes are located for the virus. And one of the main takeaways for this study is the upregulation of nuclear factor kappa beta pathways, and is the negative regulators. And this includes NF-kB-IZ, IA-ID, and TNF-AIP3. The inhibition of this pathway is important for the virus so that it can prevent excessive inflammation, to make sure that the lung is not too damaged for its proliferation. The second takeaway is the absorption of T cell markers. So as you can see here, so the PDL1 and PDL2 are the two immune suppressive transmembrane protein that interacts with the PD-1 receptor of T cells and causing T cell exhaustions. So as you can see, they are both upregulated here. So if you look at the right-hand side, which is an IPA, the results of IPA, so the ingenuity pathway analysis, we found that for the first one, there is probably too small to read, but it says T cell exhaustion signaling pathway. As we can see, it is activated in both MOC versus GFP plus cells and GFP minus versus GFP plus cells. Okay, I only realized that I haven't been standing at the microphone. I hope you guys have been hearing me fine.
Yeah.
Okay. Yeah, so that is kind of like the takeaway. And I also just want to reiterate, like for people who are interested in the details, so using the results of this study, we further look at the distribution of transcripts on lung tissues sections for pigs that are infected with PERS, with the RNA in situ hybridization technique. So it's all in the paper. I'm not showing it here, for the sake of time. And yeah, so feel free to go have a read at our paper, and then you can find all the details if you're interested. And to kind of summarize, so there's significant overlapping of gene expression profile of the bystander and PERS infected cells, and the activation of T cell exhaustion pathways and the NF-kB signaling pathways. And there is also the unique upregulation of the negative regulators of NF-kB pathways in the infected cells compared to the mock cells and the bystander cells. And we think that what is most important is that this leads to the suboptimal adaptive immune responses, which help the virus to establish persistent infection in the host so that it might then shed the virus to non-infected pigs in the future. And then it also kind of helps us understand better how the immunopathogenesis and how the virus evades the host immune system. And I would like to acknowledge everybody on the list and the funding agencies. Thank you.